Okay, so good evening, everyone. So let's wait for 2 to 3 minutes and then I will start today's session. Okay, so I hope all of you have gone through the week two lectures. हेलो हाँ बोलो उमेश मैं रूम पर हूँ लैब में जोफिशा थी लेकिन अभी पता नहीं देखना पड़ेगा गए बस जाके देख सकते हो कोई है कि नहीं गए थे क्या ओ, ये आज सब ना जल्दी निकल गया तो पता नहीं शायद होंगे नहीं ठीक है ठीक है प्लेट रीडर यूज करना था क्या प्लेट ओ अच्छा अच्छा तो मैं वैसे 9 बजे ठीक है हाँ वैसे मैं 9:30 बजे जाऊंगा लैब तो एक बार हाँ जोफिशा हाँ नहीं तो मैं पहुंच के बताऊंगा ठीक है okay so I will start today's session so I'll share the screen. Okay so I hope the screen is visible, my voice is also audible. Okay good evening everyone. I am Akash Jyoti Pathak. I am a PMRF, one of the PMRF TAs for this NPTEL course Molecular Biology, and I'm a PhD scholar in the Department of Biosciences at IIT Bombay. Uh, and I'm working in a structural biology lab. So let's start with today's session. So can anyone summarize what all has been covered in week two? Uh, so if anyone can recall. Yeah, so basically this is the slide summarizing what has been covered. We started with carbohydrate metabolism, and in carbohydrate metabolism we looked into glycolysis Krebs cycle, and then we moved into beta oxidation of fatty acids, biosynthesis of amino acids, cell cycle regulation, and there was a demo on how to use. Yeah, anyone would like to say anything? Facts instrument, right? And then in the last part we also learned about apoptosis, right? The intrinsic pathway or the mitochondrial pathway and the extrinsic pathway. Okay. So what we will do in today's session is we will try to cover the important points which has been covered in week two, and also we will solve a few sample questions so that it helps you with the assignment in solving the assignment. And also if any one of you has any doubt in week two, any part of week two, then we can also discuss that portion or any other topic that was covered in week one. We can also take up those parts as well. Okay. Okay, so let's start with today's session. Um, So yeah, first coming to glycolysis. So glycolysis pathway, I think most of you have read about glycolysis because anyone who had biology in class 11th and 12th must have read the glycolysis pathway. So it is one of the fundamental pathway for breakdown of glucose, right? So glucose metabolism, how glucose metabolism takes place inside the cell, inside an eukaryotic cell is or the pathway by which a glucose is broken down into pyruvate, right? So what is glycolysis? So glycolysis is a metabolic pathway where glucose is broken down into two molecules of pyruvate, right? And there is a net gain of two molecules of ATP. So it is a so it takes place first of all it takes place in the cytosol, right? So it takes place in the cytosol, and it is an initial stage in the breakdown of glucose. So I hope I'm audible, right? Okay, so if any one of you have any doubts, then you can unmute yourself or you can write in the chat section also. Okay, okay. Yeah, so as I was discussing, so glycolysis, it takes place in the cytosol and it is a 10 step metabolic pathway, right? So how many steps are involved in glycolysis? There are 10 steps which are involved, and through these 10 steps, the glucose molecule is broken down into two molecules of pyruvate, and it is a universal pathway. Why it is a universal pathway? It is a universal pathway because it takes place in both because it takes place in all living organisms, in starting from prokaryotes, eukaryotes, all living organisms have this glycolysis takes place, right? And also, this pathway takes place in both aerobic and anaerobic conditions. Okay. Uh, so, uh, so glucose, you see, so if you look at this pathway, which is shown in this right hand side, so here this glucose molecule is sequentially broken down, and it is sequentially converted into a couple of intermediates, and through these intermediates, ultimately what is produced? Pyruvate molecule. So pyruvate is a 3 carbon compound, and glucose is a 6 carbon compound. So this 6 carbon compound is broken down into two molecules of this pyruvate, right? Okay, so glucose is broken down into two molecules of pyruvate. So pyruvate is a 3 carbon compound, and the net gain, what is the net gain of ATP molecules? So per molecule of glucose, there is a net gain of two ATP molecules, right? If you look at this pathway, you can see that. This pathway can be divided into two phases, right? One is the preparative phase and another is the payoff phase. So the in the preparative phase, what happens is the ATP is required. ATP is required for the generation of high energy intermediates, right? So here you see in this preparatory phase, in the preparatory phase, you see there are two steps: the first step and the third step. In the first step and the third step, there is utilization of two molecules of ATP, and then this preparatory phase starts with glucose molecule, and ultimately through this series of intermediates, ultimately what do we get? We get two molecules of glyceraldehyde 3 phosphate. We get two molecules of glyceraldehyde 3 phosphate, and then this glyceraldehyde 3 phosphate is glyceraldehyde 3 phosphate moves into the payoff phase. So why is it called payoff phase? Because in this phase the ATP molecules uh, are now liberated, or ATP molecules are generated in the preparatory phase. So these high energy intermediates which were produced in this preparatory phase, these are now broken down, and we get ATP molecules. Okay, so in the payoff phase, so the preparatory phase, what is happening? The ATP is used for the conversion of glucose to two molecules of triose phosphate. Okay, so two molecules of triose phosphate. This is triose phosphate is actually glyceraldehyde 3 phosphate, and in the payoff phase, the glyceraldehyde 3 phosphate is converted into two molecules of pyruvate. Okay, so overall reaction, if you can write the overall reaction, then what is it? It is glucose plus 2 NAD plus 2 ADP plus 2 PI is giving me two pyruvate molecules, two ATP, two water molecule, two NADH, and two H plus. So couple of things you have to remember about the glycolysis pathways. The first, there are these enzymes, few enzymes you have to remember that the first enzyme which is used for the conversion of glucose to glucose phosphate is hexokinase, right? And then which are the steps in which ATP is required. So for the during the conversion of glucose to glucose 6 phosphate, ATP is required. Then during the conversion of fructose 6 phosphate to fructose 1 6 phosphate bisphosphate, there is another utilization of ATP molecule. Then the steps where NADH is produced. So there is only one step where NADH is produced. This three glyceraldehyde 3 phosphate getting converted into 1,3 bisphosphoglycerate. So in this step, the NADH molecule is generated. Okay, so because there are this dihydroxyacetone, this also converts into glyceraldehyde 3 phosphate. There are then so two molecules here, two molecules of glyceraldehyde 3 phosphate will give me two molecules. So everything here will be multiplied by two. Here we will, because there are two molecules of glyceraldehyde 3 phosphate, we will get two molecules of 1,3 bisphosphoglycerate. There will be two molecules of NADH, two molecules of ATP. Okay, so everything will be multiplied by a factor of two here. So the net energy calculation, as was also discussed in the lecture. So if you do the net energy calculation, then you can see here that there are how many ATPs are generated in this payoff phase. There are four ATPs generated. One is when a 1,3 bisphosphoglycerate converts into 3 phosphoglycerate, and another is when phosphoenol pyruvate converts into pyruvate. So there are four molecules of ATP generated, and there are two molecules of ATP utilized. So the net ATP that is generated by the process of glycolysis is two ATP, right? There are two ATP molecules that are generated, and how many NADH do we get from glycolysis? We get two molecules of NADH, and as we had learned in the last lecture, that this NADH is a reducing equivalents. So when these will be oxidized to NAD plus, then through the electron transport chain or of oxidative phosphorylation, what we will get? So two molecules of NADH will give me, one molecule of NADH actually gives me three ATP molecules, right? So we will get six ATP, six ATP molecules. So total ATP molecules that are generated through glycolysis are 8 ATP. Okay, so six ATP molecules are generated through oxidative phosphorylation, and two ATP molecules are generated through substrate level phosphorylation. Okay, so net ATP molecule generated are 8 ATP. Okay, so these points are clear, right? Now there is one another important enzyme, phosphofructokinase 1. This is another important enzyme because uh, this is also known as the one of the regulatory steps for the control of glycolysis, and also it is known as the pacemaker enzyme of glycolysis. So this phosphofructokinase 1 is an important enzyme which is required for this conversion of fructose 6 phosphate to fructose 1 6 bisphosphate. We will learn about this enzyme in the subsequent slides, where we will understand how this enzyme is regulated. Okay, or what are the different substrate which inhibits or activates this enzyme. Okay. Okay, so we will continue and move further ahead. Okay, so now one more concept which was discovered is regulation of glycolysis. So how is the glycolysis pathway regulated? So the glycolysis pathway is regulated at three main steps. One is this at step number 1, step number 3, and step number 10. So these are the three steps where glycolysis is regulated. And if you see here, then one point to note over here is that all these steps, in all these steps, the reaction it is an irreversible reaction. All these steps are irreversible reactions, and so uh, the they are regulated at these three steps. One is this hexokinase, another is phosphofructokinase 1, and another is pyruvate kinase. Okay. Hmm. Pyruvate kinase. So at three irreversible reactions catalyzed by, so the regulation of glycolysis that takes place at three irreversible reactions which are catalyzed by hexokinase, phosphofructokinase 1 and pyruvate kinase. So, so one thing is, one very basic thing is that glycolysis will only takes place when the ATP is required, right? Or when there is a deficiency of ATP in our body. So if there is a lot of ATP molecules in the cell, then the cell would no longer require the glycolysis pathway. So in this case, the glycolysis pathway will be inhibited. So high ATP. So this is a very basic thing, right? So high ATP will obviously inhibit reactions 3 and reaction 10. So this one. So ATP. Should you see this here? So this is one of the figure which has been taken from this. Um, I took it from Cooper, I guess. So Cooper book. So the ATP molecule, what will it do? It inhibits the phosphofructokinase, and it will also ATP also inhibits the pyruvate kinase enzyme. Also, it inhibits the pyruvate kinase enzyme. Uh, Now, another enzyme which is phosphofructokinase 1. This phosphofructokinase 1 is allosterically activated by fructose 2,6 bisphosphate. So so what happens is in this glycolysis step, when glucose first in the first step glucose is converted to glucose 6 phosphate, then glucose 6 phosphate is converted into fructose 6 phosphate. So in the fructose 6 phosphate then can further be converted into fructose 2,6 bisphosphate by the enzyme phosphofructokinase 2. So this fructose 2,6 bisphosphate, it is one of these enzymes which is also involved in another metabolic pathway in gluconeogenesis and all other pathway. I'll not go into too much detail. So this fructose 2,6 bisphosphate, it activates phosphofructokinase 1 by allosterically. So this is an enzyme phosphofructokinase 1, and this fructose 2,6 bisphosphate, it binds at an allosteric site to this enzyme phosphofructokinase 1. Okay, and another one the basic thing is that as we all know about feedback mechanism, right? So if the substrate or if the product is in very high concentration, then what will it do? It will inhibit the enzyme. Okay, so it will inhibit the enzyme so that the pathway can be inhibited. So glucose, when it is converted into glucose 6 phosphate, this glucose 6 phosphate, this product will inhibit the enzyme hexokinase, so that this pathway is inhibited, or this pathway no longer moves forward. So it will act as a signal that will that can stop this pathway, so that the energy can be conserved, or if ATP is not required, then this pathway will not take place, right? So hexokinase is inhibited by glucose 6 phosphate, pyruvate kinase is inhibited by ATP and activated by fructose 1,6 bisphosphate. So fructose 1,6 bisphosphate also activates the pyruvate kinase enzyme. Now, pyruvate kinase is this concept was also discussed. So it is active in the dephosphorylated state and inactive in the phosphorylated state. Okay. Now there are several other metabolic intermediates which regulate this pathway. So acetyl CoA. One is acetyl CoA, which is produced in the citric acid cycle. So this also inhibits the pyruvate kinase. So if glucose breakdown is, if ATP is no longer required, then this path then the acetyl CoA will what will it do? Because glucose is no longer required, this TCA cycle is also not required. So this acetyl CoA will inhibit the pyruvate kinase and prevent the formation of pyruvate. Okay. So you can remember this chart. So first of all, the ATP. What is ATP inhibiting? ATP will inhibit the enzyme which is involved in step 3. That is phosphofructokinase 1. It will inhibit phosphofructokinase 1, and it will also inhibit pyruvate kinase. Okay. It will also inhibit pyruvate kinase. Then glucose 6 phosphate will inhibit hexokinase and fructose. then fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate, it will activate pyruvate kinase. Okay, it will activate pyruvate kinase. So these are the points you need to understand. Okay. So these arrows which are shown in red, so fructose, these are activating the enzyme. Fructose 1,6 bisphosphate is activating pyruvate kinase. ATP is inhibiting pyruvate kinase. ATP is also inhibiting phosphofructokinase 1. And AMP, ADP, AMP, all of these will, they are these molecules will, because if ADP is in high concentration and ATP is in low concentration, then it will signal the enzyme to activate the pathway. So AMP, ADP, all these will activate the enzymes in the which are in the involved in the regulation of glycolysis. Okay. So now answer this question: in eukaryotes, the enzymes for glycolysis and Krebs cycle are found in. Okay, so I didn't discuss Krebs cycle, but I think I discussed it in the last lecture when we were discussing about mitochondria. So that time I had discussed about Krebs cycle also. So this is a very basic question. Just now we had learned that glycolysis will take place where? Glycolysis takes place in the cytoplasm, or it takes place in the cytosol. And as we had looked in the last lecture, that Krebs cycle is takes place in the matrix of the mitochondria. The matrix of the mitochondria has all the enzymes that are required for the Krebs cycle. Okay, so the answer to this question is option A, cytosol and no, not option A. Cytosol and Golgi apparatus, this is wrong. Inner mitochondrial membrane and endoplasmic reticulum, wrong. Option number D, right? Cytosol and mitochondrial matrix respectively. Okay. Option D. Okay, correct. Okay, so now moving to the next question, the precise biochemical role of hexokinase in committing glucose to the glycolytic pathway. So, what is hexokinase? So, hexokinase is an enzyme which is involved in the first step of glycolysis, right? So, hexokinase converts glucose to glucose 6 phosphate. This hexokinase enzyme, it is mainly present in the muscles, and there is an isoform of this enzyme, glucokinase, which is present in the liver. Okay, so glucose is converted into glucose 6 phosphate. So what is been asked in this question is, so what is the biochemical role of hexokinase in committing glucose to the glycolytic pathway? So what is the function of hexokinase that you have to answer in this question? So option A is, it establishes a metabolic barrier preventing glucose from entering glycolysis. It forms a stable enzymatic complex with glucose, ensuring metabolic integrity. So this first statement, it establishes a metabolic barrier, preventing glucose from entering glycolysis. This is wrong, right? Why will it form a metabolic barrier? The glucose has to enter the cytosol, or glucose must be present in the cytosol for the glycolysis to occur. So this is wrong. It forms a stable enzymatic complex with glucose, ensuring metabolic integrity. So there is no complex that is formed between glucose and hexokinase. Hexokinase is an enzyme which will convert this substrate into product, right? So this is also wrong. Next, it initiates a cascade of biochemical reactions, irreversibly dedicating glucose to glycolysis. So, so this is true. This statement is true, and the option D, it finally regulates ATP production. So in this step, ATP production does not take place, right? So ATP is utilized here. ATP is utilized here, and ADP is formed. So answer to this question is option number C, right? Okay, yeah, all of you answered it correctly. Option number C. Uh, yeah, so look at the, let's read the detailed solution once. So glucose is phosphorylated by glucokinase in the liver or hexokinase in other tissues. I don't know. In other tissues, the phosphorylation reaction of glucose to produce glucose 6 phosphate marks the molecule for glycolysis. Phosphate group of so in this step, what happens? The phosphate group of ATP is transferred to glucose. So the phosphate group which is there in ATP, it is transferred to this the carbon number 6 of the glucose molecule, and the glucose 6 phosphate is formed. Okay. And another thing about this glucose 6 phosphate, that once glucose is converted into glucose 6 phosphate, then this has to move on further, because once glucose 6 phosphate is formed, it cannot move out of the cell, because there are no phosphate transporters which are present on the cell membrane which will allow the glucose 6 phosphate to move out. Okay, so that is why the formation of glucose 6 phosphate commits the glucose to the glycolytic pathway. Okay. Okay, I don't know how. I forgot to add the animation. So let's. This is another question. Glycolysis is inhibited when? So, so when will it be inhibited? Just now we learned about the regulation of glycolysis, right? So can anyone explain what did you understand about the regulation of glycolysis? What all will inhibit glycolysis and what will activate glycolysis? So inhibition will take place when ATP molecules are more, right? So if ATP molecules or the ATP by ADP ratio is high. So if you look at the ATP by ADP ratio, if the ATP by ADP ratio is very high, it is greater than 1, then the glycolysis will be inhibited. And another so these are high energy molecules, ATP. Then second, if NADH by NAD plus ratio, if NADH by NAD plus ratio, if this is also high, then ratio very high, then glycolysis will be inhibited. And third, if the product that is formed like glucose 6 phosphate will inhibit the hexokinase enzyme. So these are the general points by which glycolysis will be inhibited. So the first point, if you read the first statement, ATP by ADP ratio is low. So if ATP by ADP ratio is low, then glycolysis will remain turned on, or it will the metabolic pathway will take place, right? Now concentration of fructose 2,6 bisphosphate is low. So fructose 2,6 bisphosphate, where did we learn about fructose 2,6 bisphosphate? So just now we had seen in this in step number 3, right? In step number 3, when this glucose 6 phosphate is getting converted into fructose 6 phosphate, and then fructose 6 phosphate. So yeah, so this pathway. So this fructose 6 phosphate. So this pathway, fructose 6 phosphate, and then fructose 6 phosphate can be converted into fructose 2,6 bisphosphate. So fructose 2,6 bisphosphate actually it activates the glycolysis pathway, because it activates the enzyme phosphofructokinase 1, and it will promote glycolysis to take place. Okay. So this statement, concentration of fructose 2,6 bisphosphate is low, this will actually activate glycolysis. Is If it is low, then what will happen? If it is low, then glycolysis will be, yeah, glycolysis will be inhibited, right? Why will it be inhibited? Because this fructose 2,6 bisphosphate will inhibit the enzyme phosphofructokinase 1. right? Or if it is low, then this phosphofructokinase 1 enzyme cannot be activated. Okay, so this actually, this can B can also be answer of this question. Concentration, if concentration of fructose 2,6 bisphosphate is low, then glycolysis will be inhibited. Okay, and but if there is only one answer correct, then you can go for option number D. Concentration of sugar in the cell is high, right? Because if glucose is high in the cell, then there is no need of uh, then the glycolysis will be inhibited, right? Hmm. Yes. Option number D is actually wrong, right? Yes, you said it correctly. So if glucose is in very high concentration in the cell, then glycolysis must take place, right? Isn't it? So if glucose is in very high concentration, then what will the cell do? It will try to break down the glucose into pyruvate molecules, right? So the answer should be option number B. The concentration of fructose 2,6 bisphosphate is low. Okay, there has been an error in this. I will change this. I will make the changes. So is it clear now? Asking. Is it clear? Okay. Okay. So remember that fructose 2,6 bisphosphate, fructose. So here you can see in this figure. So fructose 2,6 bisphosphate, what it does, it activates the allosterically activates the enzyme phosphofructokinase 1, and therefore the glycolysis pathway will move further. And if fructose 2,6 bisphosphate is in low concentration, then this fructose phosphofructokinase 1 will not be activated, and hence the glycolysis pathway will be inhibited. So answer will be option number B. Okay. Now moving to Krebs cycle. Now this also you have learned in class 12, so I'll just quickly go through this. So in Krebs cycle, first of all, this Krebs cycle takes place in the matrix of the mitochondria, right? As we had learned in the last class. It takes place in the matrix of the mitochondria, and so in Krebs cycle, what happens, the pyruvate molecules which are formed in the cytosol will move into the mitochondria, and this pyruvate molecules will further be broken down and will be further oxidized to give carbon dioxide and water uh, via this pathway. Okay, so one thing about this Krebs cycle is it will only take place in aerobic conditions. So why? Can anyone explain why this Krebs cycle takes place only in aerobic conditions and not anaerobic conditions? Yes. Oxygen. Yeah. Yeah. Correct. Partially it is correct. Some you want to add something more? So can you elaborate it further? So basically, yes, you are correct. So what happens? This NADH, and the FADH2, which are produced, which are produced in this pathway, they will move to the electron transport chain, right? And in the electron transport chain, the electron that will be produced, that will be accepted by oxygen, and water will be produced. So and these NADH will again be reduce, or it will be oxidized back to NAD plus. So these NAD plus and FAD FAD plus, these are actually substrates. There are they are substrates for several reactions in the Krebs cycle. So if these are not there, NAD plus and FAD plus, or if the electron transport chain does not take place, if there is no oxygen, then we will not get these molecules, and hence Krebs cycle will not take place. Okay. Okay, so when I will discuss the pathway in detail, then you will understand. So just remember now that it takes place only in aerobic conditions, unlike glycolysis, which can take place in both aerobic and anaerobic condition. And it is also not an universal pathway because it takes place only in like in aerobic organisms, or in which aerobic respiration takes place in those organisms. Okay, so pyruvate is transported to mitochondria and is converted into acetyl CoA by which enzyme? By pyruvate dehydrogenase, right? So in the first step, what happens? The pyruvate which was produced during glycolysis, it converts into acetyl CoA, and then the acetyl CoA will move into the Krebs cycle, right? It will acetyl CoA and oxaloacetate will combine to give a 6 carbon compound, which is citric acid, right? So this is the first step of Krebs cycle. So acetyl CoA combines with oxaloacetic acid. Oxaloacetic acid is a 4 carbon compound, and it results in the formation of a 6 carbon compound, which is citric acid. So the first product of this cycle is citric acid. That is why this is also known as citric acid cycle, or this is a or it is also known as tricarboxylic acid cycle. So if you see the structure of citric acid, then you will see there are 3 carboxyl groups that are present in citric acid. So that is why it is also known as TCA cycle. Okay, TCA cycle. So the complete breakdown of glucose by glycolysis and citric acid cycle is 38 molecules of ATP. So compared to the 2 molecules of ATP which were getting generated, or 8 molecules of ATP if we also consider the oxidative phosphorylation, then as compared to that, from Krebs cycle, we are getting an yield of 38 molecules of ATP. And the citric acid cycle completes the oxidation of glucose to 6 molecules of carbon dioxide. Okay, so since C6H12O6, right? So we will get 6 carbon compound. From 6 carbon compound, we will get 6 molecules of carbon dioxide. 4 molecules are obtained directly from each glucose molecule. Okay, so we will see this, which are the steps where ATP molecules are generated. Uh, there are 2 molecules of ATP which are generated by substrate level phosphorylation, 2 molecules of FADH2, and 6 molecules of NADH. So as we know that 2 molecules of FADH2, 1 molecule of FADH2 gives us how many ATP? How many ATP are generated from oxidation of 1 molecule of FAD? Can you know? Two. Yes, two. Okay, so since 2 molecules of FADH2 are produced, so we will get 2 into 2, 4 ATP, right? And there are 6 molecules of NADH that are generated. 1 molecule of NADH gives 3 molecules of ATP. So 6 molecules will give me 18 ATP molecules. Okay, so 18 plus 4 plus 2. Okay, so how many ATP did you get? 18 plus 6. And also during the conversion of pyruvate to acetyl CoA, if we consider that also. So during the conversion of pyruvate to acetyl CoA, 24 हो रहा है ना? Yes, 24. Okay, 24. Okay, and then 2 molecules of ATP to FADH2 and 6 molecules of NADH are obtained from the Krebs cycle, right? And then actually you are getting 13 over here. So plus, if you add the number of ATP molecules which are generated in the glycolysis pathway, so that if you add 8, and then during the conversion of pyruvate to acetyl CoA, also you get how many molecules of NADH? 2 molecules of NADH are generated, right? So from 2 molecules of NADH, you will again get 6. So if you add all these, then you will get 13 ATP molecules, right? Is it clear now? So eight ATP molecules from glycolysis and six during the conversion of pyruvate to acetyl CoA. This actually generates two NADH molecules also. So through the oxidative phosphorylation, if when this NADH will be reduced and it will move into the electron transport chain, you will get 6 ATP from this reaction also. So total you will get 38 ATP. So the energy derived from breakdown of glucose comes from the reoxidation of NADH and FADH2 with their electrons being transferred through the electron transport chain to reduce oxygen to water. Okay, yeah, so this NADH and FADH2 that are getting produced during the Krebs cycle and glycolysis, so these will again move into the electron transport chain and all the ATP molecules will be generated. Okay, so the overall reaction, if you want to uh, know the overall reaction, what is taking place in Krebs cycle is the acetyl CoA is giving me two molecules of carbon dioxide, okay, two molecules, 3 NADH, 3H plus, FADH2 plus GTP plus CoA. Okay. Hmm. Hmm. CO2. Uh, yeah, it should be actually pyruvate, right? Or so what is this? This statement has been written in a different way actually. So the citric acid cycle completes the oxidation of glucose. So once this citric acid cycle takes place, the complete oxidation of glucose takes place. That is what it is trying to say. So so from overall from this overall starting from glucose and ultimately ending in carbon dioxide. So overall you will get six molecules of carbon dioxide. It is just not, it is not talking just about the citric acid cycle. Okay, is it clear now? Huh. So in that way it completes the oxidation of glucose. So till glycolysis the glucose was not completely oxidized, right? It was partially oxidized to pyruvate molecules. Now citric acid cycle, what will it do? It will complete this oxidation process. Okay, that. Hmm. Hmm. So yeah, this is actually this they have confused. This four molecules of ATP. This is with respect to citric acid cycle, right? Isn't it? So first, let's look at the cycle. Okay. So acetyl CoA. So this acetyl CoA. So what are the steps? You are getting one over here GTP molecule, and how many NADH you are getting? Two NADH and one FADH2, right? Two NADH and one FADH2, three NADH actually. In this step, when isocitrate is getting converted into alpha ketoglutarate, and in this step, alpha ketoglutarate getting converted into succinyl CoA, and malate getting converted into oxaloacetate. So there are three steps where NADH are produced, right? And there are two. There is one step where FADH2 is produced, and there is another one step where GTP is produced. Okay. No. It is not required. You don't have to remember all the structures of these intermediates. So just a few steps where this NADH molecules are getting generated, and the steps where ATP is generated. So in this step, if you remember, it will be good isocitrate to alpha ketoglutarate and alpha ketoglutarate to succinyl CoA. In these two steps, NADH is generated, and then again in this step, malate to oxaloacetate. Okay. So actually, what happens is this glucose is giving me two molecules of pyruvate, right? Glucose is giving me two molecules of pyruvate. Then from two molecules of pyruvate, we will get two molecules of acetyl CoA, right? And then from two molecules of acetyl CoA, this entire pathway will take place twice, right? All these will be in two in twice number. Okay. So for complete oxidation of glucose, since there are two molecules of acetyl CoA that are generated, so everything will be this pathway will take place twice for the complete oxidation of the glucose molecule. Right. Okay. So 480. Hmm. Yeah. Hmm. Hmm. Fa. Okay. So you're asking that when NAD plus is getting converted, it is producing NADH, and this FAD, it is getting reduced. FAD is getting reduced. It is producing FADH2. Why not FADH plus H? That is what you're asking, right? Okay. So for this, actually, for conversion of this FAD to FADH2, uh, the there are the enzyme that would be involved here would require two H plus molecules. So this FAD will always, when the reduction of FAD takes place, it always produces FADH2. While this NAD, when it is reduced, it always produces NADH. So the enzymes which are involved, they will help in this. They will actually convert this FAD to FADH2. Okay. So that is one of the chemical reactions. So specific reason as to why it is FADH2 and not FADH. That regarding that thing, I have to see if I can if there is any specific answer to that. I will get yeah, I will see, and if I will see if I can get any answers to that question. Okay, but I do not know actually why FADH2 and why not FADH, or do I have to look? Okay, now in the so regulation of Krebs cycle, also it will take place at two levels. So in the first step, when pyruvate is getting converted into acetyl CoA, in this step the pyruvate dehydrogenase complex that is there. So in this step, there is one regulation of Krebs cycle that takes place, and then there are three enzymes. There are three main enzymes. One is this isocitrate dehydrogenase, which converts isocitrate to alpha ketoglutarate. So whenever you see, whenever there is NADH or FADH produced, then the enzyme which is present is named as dehydrogenase. Uh, and when there is any ATP molecule produced, or it is used, so any phosphorylation transfer reactions taking place, then the enzyme is named as kinase, right? So in these steps, remember these steps. So if you can remember the steps where the regulation is taking place, then it would be good. Okay, so isocitrate to alpha ketoglutarate, there is one NADH molecule that is generated. Okay, and then alpha ketoglutarate to succinyl CoA, there is again NADH that is generated. So three enzymes, namely citrate synthase also. In the citrate synthase, the this oxaloacetate combines with acetyl CoA and citrate molecule is produced. So that is one step where regulation takes place. Then isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. These are the three steps where Krebs cycle is regulated. And another is when the pyruvate is converted into acetyl CoA. Okay, at this step also the Krebs cycle is regulated. So citrate synthase. So this is a figure I've taken from Leninger, I think. These the arrows which are marked in green, these what will these do? These substrate will activate the enzyme pyruvate dehydrogenase complex. So similar to what we just saw in glycolysis also. So AMP, NAD plus, all these what will do? These molecules will activate the enzyme, right? Similarly here also ADP. So if ATP, NADH, if these are in high concentration, then there is no need for this cycle to take place. So these will inhibit the enzyme. Okay. Then another thing you remember one more important point is that calcium is one of the molecule which can activate the isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase complex. So other than the normal things that we know that NAD plus will activate NAD plus, FAD plus, all these will activate the compound, activate the enzyme. One thing you can remember is calcium also activates this isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase complex. Okay. So citrate synthase is inhibited by ATP, NADH, acetyl CoA, and succinyl CoA. So acetyl uh, then isocitrate dehydrogenase is activated by ADP and inhibited by AGP and NADH. Then alpha ketoglutarate dehydrogenase is inhibited by succinyl CoA. So alpha ketoglutarate dehydrogenase, the product which is formed is succinyl CoA. So if this product is in very high concentration, then it will automatically inhibit the enzyme alpha ketoglutarate dehydrogenase. Okay. Uh, no, so not all the inhibitors you have to remember. These, I think, by the end of this course, these things will be. There will be so many more information. All these will. I hope these things will not be of much significance because anyway the course is molecular biology. So just the basic things, if you remember, then it will be enough good. So for the solving your assignments now, since you are getting each assignment, so there are fewer less portion. But when the entire syllabus will be there, then all these minor things will not come. The main important concepts, if you understand, then it will be okay. So there will be few, you can if you understand the concept. So first concept is that if there is ATP is more, NADH is more, then obviously all these enzymes will be inhibited, right? ATP, NADH, you do not need to remember ATP and NADH because they will anyway inhibit the enzyme. And then another thing is that if the product is high, so if succinyl CoA alpha ketoglutarate is getting converted into succinyl CoA, then if the product is in very high concentration, then also it will inhibit the enzyme, right? Similarly, if isocitrate to alpha glutarate conversion is taking place, then alpha ketoglutarate will act as a feedback mechanism, negative feedback mechanism, and it will inhibit isocitrate dehydrogenase. So these are some common sense. If you apply, then you can get through these questions. Okay. Now, some important things like calcium, if you these points, if you can remember, then okay. Now, so let's move now answer this question. In a cellular study, researchers manipulate the availability of oxygen to investigate its role in the Krebs cycle. If oxygen is limited, the impact on overall Krebs cycle activity. I don't know, for some reason I'm not able to set up the poll. Uh, I will I will make the changes. I will see what is the issue. I will fix the issue and I will set polls in the next tutorial. Okay. Mm, inhibition of Krebs cycle. Okay. Any other point? Any other answers? Yeah, so just now we saw that this Krebs cycle, it always takes place in. So these type of basic questions will come in your exam, not very specific questions. Okay, so very specific questions. If you have, for that you have to go through standard biochemistry books and you have to read the metabolism chapter. What are these intermediate? So just basic concepts if you understand. Okay, so yeah, just now we saw that this Krebs cycle is can only take place in aerobic condition, right? It was only taking place in aerobic condition because this reducing equivalents that are produced, that are oxidized, and the NAD plus which are generated, that are again used in this Krebs cycle. So yeah, the option number D is correct. Option number D is correct. It will lead to inhibition of the Krebs cycle. Okay, so what is the concept behind this answer? So there is no direct participation of oxygen in the Krebs cycle, right? So in the Krebs cycle, the entire pathway that I showed you just now, was there any specific reaction or any any path intermediate which was involved in oxygen? So there was no participation of oxygen, right? However, the cycle operates only under aerobic conditions. This is due to the fact that NAD plus and FAD, so NAD plus from NADH and FAD from FADH2 required for the operation of cycle can be generated in the electron transport chain only in the presence of oxygen. Therefore, citric acid cycle is strictly aerobic in contrast to glycolysis, which operates in both aerobic and anaerobic conditions. So this is clear, right? So yeah, the NAD plus and FAD, which is required for the operation of the cycle, it can only be generated in the electron transport chain. That is why the oxygen is required for the functioning of the Krebs cycle. Now, answer this question: the consequence of high concentration of NADH plus H plus on Krebs cycle. So just now we saw the regulation of Krebs cycle. Now try to answer this question: what will it do? It inhibits citrate synthase. So there is high concentration of NADH plus H plus. Okay. So high concentration of ATP, high concentration of NADH. What does it do? So it is a general thing that it will always inhibit the glycolysis or Krebs cycle, right? Option number B. Okay. Any more answer? So yeah, option number B is correct. It will not activate, right? If the concentration of NADH and H plus is high, you can eliminate this option number C and D. And also option number E is also correct for this question, right? We just now saw here. Okay, so if NADH is high, then it is inhibiting the citrate synthase, right? So you see here, so cross mark means this NADH, succinyl CoA, citrate, ATP, these molecules, they are inhibiting the enzyme. So there were three main enzymes which were regulated in the Krebs cycle, which are citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. So if you see them, then all these three enzymes, they are inhibited by the NADH, high concentration of NADH and ATP. Okay, high concentration of NADH and ATP will inhibit these three enzymes. Okay, so both option number, I don't know, there is something. Option number A and B. Okay, there is some error. Option number, yeah, no, isocitrate dehydrogenase is also inhibited. It is not shown here, but NADH will also. Option number alpha ketoglutarate. Okay, in alpha ketoglutarate. Okay, here, yeah, you are correct. In that case, ATP will not inhibit. Okay, now let's move on to beta oxidation of fatty acids. Okay, so beta oxidation of fatty acids means what? From where are we getting fatty acids? First of all, so any fat that we take through our diet, right? Any fat that we take in our diet, that gets converted into what? It gets converted into triacylglycerols and fatty acids, right? Now, how this oxidation of fatty acids take place to generate ATP, or how the metabolism of fatty acids take place, we will learn now. So the complete oxidation of fatty acids to carbon dioxide and water takes place in three stages. So first, what will happen? The oxidation of the long chain fatty acids to two carbon fragments in the form of acetyl CoA. So ultimately, what is happening over here in beta oxidation of fatty acids? So this is a fatty acid, right? Uh, palmitoyl CoA. So this palmitoyl CoA, ultimately, what happens? There are subsequent steps, and in each step, one of the acetyl CoA molecule is liberated. Okay. And so in the first step, one acetyl CoA molecule will be liberated, and now we will be left with 14 carbon compound. Then in the next step, again this acetyl CoA will be liberated, and you will get a 12 compound. And then in this way, one by one, this will take place. And in this four-step process, at each step there will be removal of two carbon fragments. Okay. Uh, so that is what it is written here. The oxidation of long chain fatty acids to two carbon fragments in the form of acetyl CoA, and the oxidation of acetyl CoA to carbon dioxide in the citric acid cycle. So this acetyl CoA that is generated in this oxidation of fatty acid, what will happen to this acetyl CoA? This acetyl CoA, this will again move into the citric acid cycle, TCA cycle. And then the TCA cycle, what will happen? This acetyl CoA will be converted into carbon dioxide and water. Okay, so transfer of electrons from the reduced electron carriers to the mitochondrial respiratory chain. The stage Yeah, so what happens in stage one? In stage one, beta oxidation is a four-step process which involves the sequential removal of two carbon fragments from the carboxyl end of the fatty acid as acetyl CoA. So why is it beta oxidation of fatty acid? Because this alpha beta bond is broken. Uh, the from the removal of the carbon fragments from the carboxyl end of the fatty acid takes place. That is why it is called beta oxidation. And the bond between the alpha and beta carbon atoms are broken. This, you see, this is the alpha. So the carbon which is attached to this carboxyl or the CO group, the CO group is called the alpha carbon, and adjacent to the alpha carbon is the beta carbon. Okay. It is named so because at the end of each of the four steps, the beta carbon at the start of the cycle is oxidized to CO. So after this four step process, what will happen? This beta carbon is getting converted into CO. Okay, at each step, and you are getting acetyl CoA. Now, what happens? So, in eukaryotes, the beta oxidation takes place in the matrix of the mitochondria. So, in the matrix of the mitochondria, the enzymes for Krebs cycles are also present, and the enzymes for beta oxidation of fatty acid is also present. So, the free acids with chain lengths which are less than 12 carbons enter the mitochondria without the help of membrane transporters. So fatty acids can have any number of carbon atoms, right? There can be 12 carbon atoms. There can be more than 12 carbon atoms. So if my fatty acid has less than 12 carbons, then the fatty acid can easily enter the mitochondria without any membrane transporters. But if there are more than, greater than, or equal to 14 carbon which are present in fatty acid, which constitute the majority of the free fatty acids obtained in the diet or released from adipose tissue. These fatty acids cannot directly pass through the mitochondrial membrane. Okay, so for this process, what happens? They must first undergo the three enzymatic reactions of the carnitine shuttle. So what happens is this fatty acids are produced in the cytosol. Now from the cytosol, they have to move into the matrix of the mitochondria. They have to move to the matrix of the mitochondria. Now now fatty acids which have a chain length greater than 14 carbon atoms, those cannot directly enter into the matrix of mitochondria. So for that process, there is a shuttle present in the mitochondria, which is called the carnitine shuttle. Okay, so we will understand this carnitine shuttle because there are a few questions which have been asked in your assignments as well from this part. So what is happening here? So if you see the fatty acid, so fatty acid, this is a what is a fatty acid? It is a So it has a hydrocarbon chain, and there is a carboxyl group, right? Fatty acid. So we will learn about the biomolecules in the next in the week 3 we will learn about different biomolecules. So it has a carboxyl group. So first what happens, this CoA molecule, this is coenzyme A. This coenzyme A is first a reaction takes place, and a thioester bond is formed. Fatty acyl CoA is formed. Okay, and which is the enzyme that is involved? It is acyl CoA synthetase. Acyl CoA synthetase is also called as acyl CoA ligase. Okay. So this enzyme helps in the formation of this fatty acyl CoA, and then this acyl CoA synthesis catalyze the formation of acyl CoA synthase. What it does, so try to remember this first enzyme. Okay, so this acyl CoA synthase, this is an important enzyme. It catalyzes the formation of a thioester linkage between the fatty acid carboxyl group and the thiol group of coenzyme A. So so this is the fatty acyl carboxyl group. So the fatty acyl, between the fatty acyl carboxyl group and the if you look at the structure of coenzyme A, there is a sulfur atom. So this there is a thiol group. So between these two, the bond which is formed is called a thioester linkage. So once this, okay, now what will happen? So now what happens? So you see, first what happened in the cytosol. In the cytosol, what happened? This fatty acid got converted into fatty acyl CoA by the help of which enzyme? Fatty acyl CoA synthetase or fatty acyl CoA synthase. Okay, fatty acyl CoA synthase. The first we are getting fatty acyl CoA. Now this fatty acyl CoA, so this is directly from the lecture slide. So it is converted into fatty acyl carnitine derivative in a reaction named transesterification by the enzyme carnitine acyl transferase 1. So there is an enzyme carnitine acyl transferase 1. What it does, it transfers the carnitine group to the fatty acyl CoA. So now you see this S CoA, which was there, this group has been replaced by a carnitine molecule. And who does this? There is this enzyme which is carnitine acyl transferase 1. So this enzyme catalyzes the transfer of this carnitine molecule to the to the CO group. Okay, now now where is this carnitine acyl transferase present? Carnitine acyl transferase is present in the outer mitochondrial membrane. Okay, so as we had learned in the when we were talking about mitochondria, that the inner membrane is impermeable to small molecules and various other molecules, other small molecules, other molecules. Okay, so that is why there is this translocase, this acyl carnitine translocase. This acyl carnitine translocase is present in the inner mitochondrial membrane, and this translocase transfers this carnitine from the intermembrane. So from this outer mitochondrial membrane, it will move into the matrix of the matrix of the mitochondria. And now when it moves into the matrix of the mitochondria towards this matrix side of the inner membrane, there is another enzyme which is carnitine acyl transferase 2. So what it does, it uh, it uh, ca- it replaces the carnitine with this S CoA group, and this carnitine is again recycled. It is recycled back into the cytosol. Okay, so this is carnitine shuttle system that you need to understand. Okay, so I hope this is clear. So first, what happens? So this derivative is translocated to the mitochondrial matrix. So this fatty acyl CoA. So how? So the fatty acids are there in the cytosol. So how will it move into the mitochondrial matrix? Because first it has to move into the mitochondrial matrix for the beta oxidation to take place. So for this, the fatty acid is first converted into fatty acyl CoA. Who does this? There is an enzyme acyl CoA synthase which converts this fatty acid into fatty acyl CoA. Now, in the outer mitochondrial membrane, there is an enzyme carnitine acyl transferase which replaces this S CoA with carnitine, and then now this fatty acyl CoA can move into the intermembrane space. Now, from the intermembrane space, it moves into the matrix via this channel protein. So there is this channel protein carnitine, acyl carnitine translocase. So this is the name of this translocase protein. This channel protein which transfers this carnitine molecule into the inner side of the inner inner matrix side of the inner membrane. And here, then again, what will happen? This carnitine acyl transferase 2 enzyme is present. So carnitine acyl transferase 2 will again replace this S CoA with carnitine, and this carnitine will be recycled. Okay. So that is what is the fatty acyl CoA is regenerated via carnitine acyl transferase 2 located on the matrix side of the inner mitochondrial membrane. Carnitine acyl transferase 1 was located on the outer membrane. Carnitine acyl transferase 1 is located on the matrix side of the inner mitochondrial membrane. And then what happens? This carnitine is transported back into the intermembrane space via the acyl carnitine transporter. which is then ready to participate in other reaction of activating fatty acid. Is it clear? So till now, what we understand? Yes, please. Uh, where, on the matrix side of the inner membrane, as carnitine acyl transferase 2 is present. So this resolution is 4. This is actually 2. Okay, this is 2 and this is 1. Okay, so 1 is present in the outer membrane and 2 is present on the matrix side of the inner membrane. Okay, so we had discussed in the last class, if you remember, that this inner membrane is impermeable to large molecules. Yes. So because of this, this channel protein, this is a channel protein which allows the transport of this carnitine fatty acid carnitine complex from the intermembrane space into the mitochondrial matrix. Okay. Okay. So now what happens? So if it is a 16 carbon fatty acid, so so there are multiple steps in this beta oxidation. So in the first step, we will get a 14 carbon compound and an acetyl CoA. Acetyl CoA is a 2 carbon compound. Okay, then again this C14. So there are multiple enzymes which are involved. I'm not going into too much detail, and you also don't need to remember so many enzymes and what are the different thiolase enzyme, hydratase enzyme. There are many steps involved. Okay, so just you understand the basics. So C14 will be converted into C12 and we will get again acetyl CoA. So that there will be this that 2 carbon compound. Then C12 will be converted into C10. Then C10 will be converted into C8. Okay. Then C8 will be converted into C6. C6 will be converted into C4. So how many acetyl CoA molecules you are getting from 16 carbon fatty acid? So you are getting 7 or 8. So 3, 4, 5, 6, 7, 8. You are getting 8. Okay, because this C8 will be converted into C6, then C6 to C4, and then C4 to ultimately will get only acetyl CoA. Ultimately, what you will be left with is acetyl CoA. So C16 to if it has to be completely oxidized, you will get you will get 8 molecules of acetyl CoA. So now again there is this regulation of fatty acid oxidation. There are several enzymes that I have not touched upon. You can go through the lecture. So first, this one enzyme is actually important. So glucose, it gets converted into acetyl CoA, and this acetyl CoA gets converted into is acetyl CoA carboxylase. This acetyl CoA carboxylase converts this acetyl CoA to malonyl CoA, and this malonyl CoA it inhibits the carnitine acyl transferase 1. So carnitine acyl transferase 1 was present on the outer membrane of the mitochondria, right? So this malonyl CoA actually inhibits this carnitine acyl transferase 1, so that this fatty acid oxidation can be inhibited. So this is one of the regulatory steps or the main regulatory step where this fatty acid oxidation is inhibited. Okay, so what happens? So when the diet provides a ready source of carbohydrate as well, beta oxidation of fatty acid is unnecessary. So when will you require oxidation of fatty acid? So if when the glucose levels are very low, when your diet is not giving you sufficient glucose, right? Or you are under starvation. So during starvation, all the glucose that is present in your all the glucose that was stored in your body that has been utilized. Now, what is left now? What will happen now? The lipids and the adipose tissue, all the lipids will start to oxidize. So during this step, what will happen? The beta oxidation of fatty acids will takes place. So when diet is provided, so what this line says is when the diet is provided, the beta oxidation of fatty acids is unnecessary, right? There will be no need of fatty acid oxidation because this glycolysis and citric acid cycle can provide us with the energy, and therefore this pathway is down regulated. So when will this pathway be down regulated? When there is sufficient glucose or when there is available energy present. So this acid will be down regulated. So how will it be down regulated? So it is inhibited in the very first step. This fatty acid, this malonyl CoA, is not allowed to enter the matrix of the mitochondria, because once it enters the matrix of the mitochondria, there will be several enzymes of the which are involved in beta oxidation of fatty acids. So they will start to take place one by one. So therefore, it is inhibited in the very first place, and it is not allowed to enter the matrix of the mitochondria. Okay. So that the two enzymes are key to the coordination of fatty acid metabolism. One is the acetyl CoA carboxylase. So what is acetyl CoA carboxylase? This is the first enzyme in the synthesis of fatty acid, and acetyl and carnitine acyl transferase 1, which limits the transport of. So if this carnitine acyl transferase 1, if this enzyme is inhibited, then what will happen? It will limit the transport of fatty acids into the mitochondrial matrix for beta oxidation. And so the pathway can be inhibited. Okay, is it clear? So now, so here, so actually there is too much information. So in ingestion of a high carbohydrate meal, what it does, it raises the blood glucose level and thus triggers the release of insulin. So whenever you take a high carbohydrate diet, what happens? Which enzyme is released? The insulin is released from the pancreas, right? And so when insulin is activated, what it will do? So you have to remember that when there is enough glucose, when there is enough glucose, then this beta oxidation of fatty acid will be inhibited. Okay, beta oxidation of fatty acid will be inhibited. This has to be inhibited. So this cycle has to be inhibited. So it will start with. So first, what will happen? There will be release of insulin when you take a high carbohydrate diet, and then what happens? This insulin-dependent protein phosphatase dephosphorylates acyl CoA carboxylase. So acetyl CoA carboxylase activating it. So you see here, when this acetyl CoA carboxylase is phosphorylated, it is in an inactivated state, while when it is dephosphorylated, this acetyl CoA carboxylase gets activated. Okay, so acetyl CoA now. Once it is dephosphorylated, the acetyl CoA carboxylase gets activated. Now, acetyl CoA carboxylase, what will it do? It will catalyze the formation of malonyl CoA. So, acetyl CoA carboxylase, it converts acetyl CoA to malonyl CoA, and this is the first intermediate of fatty acid synthesis. And then what it does? So, once this malonyl CoA is produced, it inhibits carnitine acyl transferase, thereby preventing fatty acid entry into the mitochondrial matrix. When blood glucose levels drop between meals, so when the blood glucose level will drop, which hormone will be released? This glucagon peptide is released, right? So glucagon peptide, what will it do? It will phosphorylate the acetyl CoA carboxylase. Once this so it will so Glucagon release activates calcium, this cyclic AMP dependent protein kinase A, which phosphorylates. So this will activate the cyclic AMP and protein kinase A, and this protein kinase A, it will subsequently, what it will do? It will phosphorylate the acetyl CoA carboxylase and make it inactive. So once this ACC is inactive, then this malonyl CoA will not be produced, and then this. So the concentration of malonyl CoA falls, the inhibition of the fatty acid entry into the mitochondria is relieved, and fatty acids enter the mitochondrial matrix and become the major fuel. Because glucagon also triggers the mobilization of fatty acids in adipose tissue, a supply of fatty acids begin arriving in the blood. Okay, so malonyl CoA is an inhibitor of this carnitine acyl transferase 1. So, and who is controlling the this malonyl CoA, or regulating the concentration of malonyl CoA? It is acetyl CoA carboxylase. So acetyl CoA carboxylase, when it is phosphorylated, then this malonyl CoA will not be produced, and when acetyl CoA carboxylase is dephosphorylated, it will remain active and it will convert this acetyl CoA to malonyl CoA. Okay. Now answer this question. The primary enzyme responsible for the activation of fatty acids before their entry into beta oxidation. So the very first step, I hope you all remember. So in the very first step, the fatty acid is first converted into fatty acyl CoA. So I was emphasizing on this point that you need to remember the fatty acyl CoA, the enzyme involved in this. So the activation of fatty acid into fatty acyl CoA is brought about by which enzyme? So this option number D. Okay. Any more answer? All of you agree with option number D? It is acyl CoA synthetase. Okay. So this fatty acid to fatty acyl CoA, this takes place in the cytosol, and after this compound has been formed, fatty acyl CoA, now this can move into the mitochondria via the carnitine shuttle system. Okay. So yes, the answer is option number D. Now answer this question. In eukaryotes, beta oxidation primarily occurs in. So this is also very easy. Mitochondria, right? Because all these enzymes of this beta oxidation, they are present in the mitochondria. Okay, so answer is option C. Now acyl CoA synthetase catalyzes the formation of dash linkage between the carboxyl group of fatty acid and the thiol group of coenzyme A. Which bond was being formed in this fatty acyl CoA? So thioester. So COS, this bond is called the thioester bond. So this OA, so this bond is called the thioester bond, and this bond is catalyzed by this acyl CoA synthetase. Okay, so this one, if you are forgetting, this one, this COSOA, this one. Okay, okay, we'll move ahead now. Now, now to answer this question, if a cell lacks functional carnitine acyltransferase 1, so the carnitine acyltransferase 1 is lacking. Okay, so this carnitine acyltransferase 1, it was present on the outer mitochondrial membrane, and it was helping in the transport of fatty acid to the mitochondrial matrix. So now answer. Okay, it will inhibit fatty acid transport into the mitochondria. Yes, answer is option B. Okay, so if this is lacking, then this fatty acid cannot be transported into the mitochondria. So, so these type of questions were there in your assignment, and if you just remember this concept of carnitine shuttle system, it is enough, because there are many other enzymes which are involved in this beta oxidation. So let's skip those. So dash is the rate limiting step for fatty acid oxidation. So what is the rate limiting step for fatty acid oxidation? Acyl CoA synthetase activation, beta hydroxyacyl CoA dehydrogenase activity, carnitine acyltransferase 1 activity, the carnitine shuttle. So rate limiting step. So which was that? Which step was if you was getting if you block that step, then this fatty acid oxidation would not take place. Which is that particular step? If does not take place, then there will be no oxidation of fatty acid. So based on that, you have to answer. Okay. Okay. Any more answer? Yes, actually, answer is option number C, because if this acid if this carnitine acyl transferase 1 is inhibited, so that's why I was discussing that malonyl CoA part in more detail, because what will happen when malonyl CoA is formed, it inhibits this carnitine acyl transferase, so that this so that in the very first place, the fatty acid will not move into the matrix of the mitochondria, and hence there will be no oxidation of fatty acid. So yes, answer is option number C. So these questions are clear, right? Okay, so I'll move ahead then. Now cell cycle, this is there is lots of information, or there is a very detailed description of cell cycle. If you want to learn, then you can refer to very standard books. But for this course, you do not need to go, just the basics of cell cycle is enough. Okay, so there are this cycline and cdk. There are several cdks which cdk activates with cycline, and how the complex is formed, which gets dissociated at which phase. So there are a lot of information, but for this course, we will just restrict ourselves to the basics of cell cycle. So you all know the cell cycle. There are four steps. This it is divided into the interphase and mitotic phase, right? So so the mitotic phase lasts for around 1 hour. If we look at the cell division in a eukaryotic cell, then the total duration for a division of a cell is approximately 24 hours. And during this cell cycle, what happens? There is four coordinated processes taking place. Starting from, so first what happens? There will be the growth of the cell. So in this growth one phase, or it is also known as the gap one phase, the cell growth take place. Then in the S phase, what happens? S phase, DNA replication takes place. And in the G2 phase, there are several proteins which are synthesized, which will facilitate the mitosis, or the division of the cell to happen. Okay, it consists of four coordinated processes: cell growth, DNA replication, distribution of the duplicated chromosome to daughter cells, and cell division. So it is divided into two basic parts. So first is one is the mitotic phase. So this one, M phase, and this G1 as G2. These together, all these stages, they are together called the interphase, right? Interphase. So interphase is the phase when the cell is preparing for division, and the actual division takes place in the mitotic phase. Okay, so what is happening in mitosis? In the mitosis, there is separation of daughter chromosome followed by cytokinesis. So the daughter chromosomes are separated, and then the cell will also divide. Once these chromosomes are separated, there will be cytokinesis, or the division of the cytoplasm. Then interphase. So in the interphase, what happens? Both cell growth and DNA replication occurs in an orderly manner in preparation for cell division. Okay. So mitosis and cytokinesis last for only about an hour. So if you look at this 24-hour period for cells, for cell division, in this 24-hour, 1 hour is only 1 hour is required for cell division, while the remaining 23 hours is required by the cell for preparing for the cell division. Okay, it is spent in the interphase. That is the period between mitosis. So interphase is a period between two successive mitosis. Okay. So now there are the what are the phases of cell cycle? So in the interphase there are three phases: G1 phase, S phase, G2 phase. So in the G1 phase, what happens? So in the G1 phase, the cell is metabolically active and it grows continuously. So this lasts for 11 hours. Uh, for this facts experiment, so if you can have a rough understanding of which phase is or a relatively longer compared to the other phases, then it will be good. Okay, so in the G1 phase, it takes place for the maximum duration of time. It takes around 11 hours for the G1 phase, and the S phase will last for 8 hours, and G2 phase lasts for 4 hours. So in the G1 phase, the cell grows and cell is metabolically active. In the S phase, the DNA replication takes place. It lasts for 8 hours. And the G2 phase, what happens? The proteins are synthesized in preparation for mitosis, and it lasts for 4 hours. So M phase is the phase where mitosis or the cell division happens. Now there is another phase which is called the G0 phase, right? You all know about the G0 phase. So cells which can divide only once, or which have differentiated terminally and cannot grow, or they cannot divide, like there are certain brain cells, which or the nerve cells, these cells move into the G0 phase. So some cells in adult animals cease division altogether, example nerve cells, and many other cells which divide only occasionally as needed to replace cells that have been lost because of injury or cell death. So these move into the G0 phase. So what happens? These cells exit G1 phase to enter a quiescent stage of the cycle called the G0, where they no longer proliferate. So in the G0 phase, the cell remains metabolically active, but it does not divide further. And how so the phases, if you remember the phases of mitosis, so who can tell the phases of mitosis? Telophase, right? Prophase, metaphase, anaphase, telophase. Okay, so these phases you can easily distinguish by looking under the microscope, right? So if you look under the microscope, you can easily distinguish, and many of you would have done this in your practicals, onion tip root cell, and you can identify the different stages of mitosis. But how will you identify the stages of interphase? So in the interface, the G1 S G2, how will you identify them? So for identifying these stages, that can only be, or these phases can only be differentiated based on the DNA content of the cell. And for this, an instrument is used, which is FACS, right? Or also it is known as flow cytometer or fluorescence associated cell sorting. So this instrument helps us in segregating the cells which are in G1 phase, S phase, and G2 phase. Okay. So if you see this figure on the right, so here what you can see, the y-axis has number of cells and this x-axis has the relative amount of DNA. So in the G1 phase, what is the amount of DNA present in the cell? It is, so if the amount of DNA present in the cell is C, then what will happen in the S phase? The amount of DNA becomes 2C, and in the G2 and M also it will, this will this 2C will remain. Okay. So this is a profile this is this is a profile that you get when you sort cells using a flow cytometer. So I think there was a demo which was given to you in the lectures. So what happens? So if you see here, so since the most of the duration of cell cycle is taken by the G1 phase, right? So G1 phase lasts for 11 hours. So that is the maximum time which a cell takes place is in G1 phase. So that is why here you see that the G1 phase has the number of cells in G1 phase are maximum. So you get a high peak over here, and then and the G2 and M phase, G2 and M phase. Uh, 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 in the G2 and M phase, it lasts for 4 plus 1 hour, so 5 hours around. Okay, so here also, here here you will get a lower peak. Okay, now in the S phase, there will be few cells which will have this 2C. There will be an intermediate between the C and 2C. That is why you see this phase between this G1 and the G2 and M phases. Okay, and in the G2 and M, all these cells will be having 2C amount of DNA. That is why this you the amount of DNA per cell is here too. Okay, give me a time. Just give me 1 minute. हेलो. हाँ सर बोलिए. अभी रूम, हाँ हाँ रूम में हूँ. ये 9 बजे खत्म हो जाएगा और ऐसे ही 20. हाँ हाँ. नहीं नहीं ठीक है, मैं जा आऊँगा लैब. आप लैब में ही हो? ठीक है हाँ. Okay, so are you able to follow, Shamiksha? What is confusing you? Oh, insulin. Okay, okay. So okay, I will go through that once more. Okay. So yeah, so you tell me how much you want. Okay, in Hindi you want. Okay, 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 okay. तो देखो अब ग्लूकोज हमें ये बीटा, ये पहले तो ये बताओ बीटा ऑक्सीडेशन ऑफ फैटी एसिड्स कब होगा बॉडी में, मतलब सेल में कब होगा? When will this beta oxidation of fatty acid take place? So beta, हाँ बोलो, स्टारवेशन में है या हाँ, तो उस टाइम जब बॉडी में ग्लूकोज अवेलेबिलिटी कम है तो उस टाइम ये चलेगा, है ना, फैटी एसिड ऑक्सीड बीटा ऑक्सीडेशन ऑफ फैटी एसिड। सो बाकी टाइम जब जरूरत नहीं है, तब मतलब जब ग्लूकोज अबंडेंट है, जब सेल में ग्लूकोज अबंडेंट है, तब ये पाथवे चलने का जरूरत नहीं है, राइट? So, at when the glucose is in high concentration inside the blood, then there is no need for this pathway. Okay, तो इस टाइम क्या होता है कि ये जो है ना, ये कार्निटिन असाइल ट्रांसफरेज वन जो एंजाइम है ना, इसी को इनहिबिट कर दिया जाता है। तो ये कैसे इनहिबिट होता है? तो इसमें दो दो मतलब देखो, एक डाइटरी कार्बोहाइड्रेट हम लोग मान लो कुछ खाया, हम लोग ने राइस चावल दावल कुछ खाया हम लोग ने, तो उस टाइम क्या हुआ, बॉडी में ब्लड ग्लूकोज लेवल्स बढ़ जाएगा बॉडी में। तो ब्लड ग्लूकोज लेवल बढ़ जाएगा तो क्या होगा, जैसे ही ब्लड ग्लूकोज लेवल बढ़ेगा तो इंसुलिन का कंसेंट्रेशन भी, इंसुलिन रिलीज होने लगेगा। तो जैसे ही इंसुलिन रिलीज होने लगता है, तो ये क्या करता है, इसमें भी और भी एंजाइम्स वगैरह है जो अल्टीमेटली क्या करता है, ये एसिड एसाइल एसिटाइल कोए कार्बोक्सिलेज जो है ना, इसको ये एक और फॉस्फेटेज एंजाइम को एक्टिवेट करता है। ठीक है, इंसुलिन क्या करते हैं, ये फॉस्फेटेज एंजाइम को एक्टिवेट करता है। अब ये फॉस्फेटेज एंजाइम जैसे ही एक्टिवेट होगा, ये क्या करेगा, ये जो फॉस्फेट ग्रुप जो इस एसाइल कोए कार्बोक्सिलेज जो है ना, इस एसिटाइल कोए कार्बोक्सिलेज, इस एंजाइम पे जो फॉस्फेट ग्रुप लगा रहता है उसको डीफॉस्फोराइलेट कर देगा। So once this dephosphorylation takes place, as soon as this dephosphorylation takes place, this acetyl-CoA carboxylase, this enzyme gets activated. ये एक्टिवेट हो गया, और जैसे ही एक्टिवेट हो जाएगा, ये एसिटाइल कोए किस में कन्वर्ट हो जाएगा, मैलोनाइल कोए। नाउ मैलोनाइल कोए क्या करता है, मैलोनाइल कोए इस कार्निटिन एसाइल ट्रांसफरेज 1 को इनहिबिट करने लगता है। ठीक है, so once this carnitine acyl transferase 1 is inhibited, then this fatty acid will can no longer move into the, it can no longer move into the matrix of the mitochondria। ये मैट्रिक्स है, समझ आया अभी? ये जब हाई मतलब जब ग्लूकोज ब्लड ग्लूकोज लेवल हाई है, तो इस पाथवे को बंद रखने का कोशिश कर रहे हैं, ये पाथवे ना चले, क्योंकि इस पाथवे भी, ये पाथवे भी अगर चलेगा जब ग्लूकोज लेवल हाई है, तो इसमें भी तो एटीपी लगेगा ना हमें, ये सारे कन्वर्शन में इतने सारे एंजाइम्स जो है एक से दूसरे में कन्वर्ट हो रहे हैं, इसमें इसमें जो एंजाइम्स लगेगा उसमें जो एटीपी रिक्वायरमेंट है वो ना लगे, इसीलिए ये पाथवे को बंद कर दिया जाता है। कौन सा? नहीं, ये सिर्फ, हाँ हाँ हाँ, ये सब जब, ये तो कंटिन्यूअस चलता रहेगा, लेकिन जब ग्लूकोज वगैरह ज्यादा होगा बॉडी में, तो तब ये बंद किया कर दिया जाता है, ये फैटी एसिड ऑक्सीडेशन पाथवे, है ना। नहीं, ये ये क्यों चलेगा, जब जब तुम्हें ग्लूकोज और ट्राइकार्बोक्सिलिक एसिड साइकिल से जब तुम्हें एटीपी जितना जरूरत है उतना मिल रहा है, कि ये चलने का क्या जरूरत है? ये सब तुम्हें तब जरूरत होता है ना, जब ये फैटी एसिड से ग्लूकोज वगैरह तब बनता है ना, जब ये ग्लूकोनियोजेनेसिस वाले प्रोसेस जो हम लोग पढ़ते हैं। ये सब कब होता है? स्टार्वेशन कंडीशंस में फैटी एसिड, फिर पाइरुवेट, फिर ये सब से जब ग्लूकोज बनने लगता है, इस प्रोसेस को हम लोग ग्लूकोनियोजेनेसिस बोलते हैं ना। तो ये ग्लूकोनियोजेनेसिस तो तभी चलता है जब ग्लूकोज कम हो, या स्टार्वेशन कंडीशंस में, या एक्सरसाइजिंग कंडीशन में, जब ग्लूकोज जितना भी बॉडी में स्टोर था वो यूटिलाइज हो जाता है, उसके बाद ये सब प्रोसेस स्टार्ट होता है। प्रेफर्ड सबस्ट्रेट फॉर एटीपी प्रोडक्शन क्या है? कार्बोहाइड्रेट होता है ना हमेशा। कार्बोहाइड्रेट। हाँ। हम्म, हाँ। तो वही जो हम लोग खाते हैं ना फैट्स, वो फैटी एसिड्स में ये सिंथेसिस होता है, तो फैटी एसिड्स में कन्वर्ट हो जाता है, एंड दिस फैटी एसिड्स रिमेन स्टोर्ड इन आवर एडिपोज टिश्यू, एंड एंड जितने भी हमारे जो भी मसल्स है इधर उधर स्टोर करके रख देते हैं। एडिपोज टिश्यू वगैरह में फैटी एसिड स्टोर करके रखते हैं, फैटी एसिड और ट्राइएसाइक्लिसरॉल मिला के लिपिड के फॉर्म में स्टोर रहता है। नहीं, 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 सिर्फ फैटी एसिड से ऐसे स्टोर होता है। कार्बोहाइड्रेट तो स्टार्च के फॉर्म में हो जाता है ना। कार्बोहाइड्रेट ऐसे नहीं स्टोर रहता है। ओके। So this point is clear, right? Phases of cell cycle. So G1 phase lasts for the maximum duration of time. That is why we get a the number of cells in the G1 phase are higher compared to the S and G2 and M phases. Okay. So cells in S phase, the minimum number of cells will be in S phase, while in the G2 and M phases there will be a more number of cells compared to the S phase. Okay. Now there are this cycle checkpoints. So just the basics of cell cycle checkpoint. So what are the checkpoints and why is it required? So when cell cycle, so this is also a way by which the cell cycle is regulated. Okay. So this is a mechanism by which the cell cycle is regulated, and it helps in, it acts as a control to check if all the substances that are required in one phase has been successfully done. Okay. So what happens? So so in each phase some molecules, some protein molecules will be synthesized, right? So in the G1 phase there will be several protein molecules, several amino acids, nucleotides, all these will be present, will be synthesized in the G1 phase. Now when the cell will move to the G1 to the S phase, there will be a checkpoint. So what will this checkpoint do? It will check whether there is availability of all these substances which are required for the replication of DNA. Okay. Now, then when the cell will move from the S to G2 phase, then there will be again a checkpoint which will check whether there is any DNA damage, whether there is any error in DNA replication, whether everything has been synthesized correctly. And once it finds that everything is okay, then the cell is allowed to move from the S to G2 phase. Okay. Then similarly, when the cell moves from the G2 to M phase, then again the cell will check for whether all the substances which are required for mitosis is there in my cell or not. So here first it will check, is all the DNA replicated, is the environment favorable, are the growth factors that are required for division, are they present or not. So once it checks for all these, then the cell is allowed to transition from the G2 to M phase. Okay. So enters mitosis. Then again in the M phase, what happens? In the M phase there is a stage in the mitosis which is known as anaphase. Okay. So in the metaphase to anaphase stage, what happens? In the metaphase to anaphase stage, there is a checkpoint which is known as the spindle assembly checkpoint. Okay, spindle assembly checkpoint. So in this checkpoint, what the cell checks for is whether it checks whether all the chromosomes have aligned in the equatorial plane, whether all the chromosomes have aligned in the equatorial plane, and whether all the chromosomes have been or whether all of them are associated with the spindle fiber. So in the metaphase, what happens? The chromosomes align in the equatorial plane, and then in the anaphase, the chromatids get separated. So in the spindle assembly checkpoint, what does the cell check for? It checks whether all the chromosomes are aligned in the equatorial plane and whether all of these chromosomes have been attached to the spindle fiber. So so this is what, yeah. So it will check are all chromosomes attached to the spindle, and once it checks that yes, it is everything is okay, then only the cell is allowed to transition from the metaphase to anaphase. Okay. So this trigger anaphase and proceed to cytokinesis. So there are several checkpoints which are present in the in different phases of the cell cycle. Okay. So there lot of details can be found in different books in Alberts, and in standard books you can find details, but just the basic is required for this course. So there are several cell cycle checkpoints called DNA damage checkpoints, which function to ensure that the damaged DNA is not replicated and passed on to daughter cell. So there are basically three main checkpoints. One is the G1S checkpoint, second is the G2M checkpoint, and another is the M phase. So in the G1S checkpoint, what does the cell do? In the G1S checkpoint, the G1S checkpoint allows the repair of any damage to take place before entering the S phase. So if there is any damaged DNA, or if there is any error, if the cell finds that there is any error in this G1 phase, then the cell will not be allowed to proceed to the next phase. Okay, so all these checks are done. Uh, so is the environment favorable? Can the cell and proceed to the S phase? If are there any damage, or are there any DNA damage present? So all these will be checked in the G1S checkpoint. Then in the G2M checkpoint, what happens? That it prevents initiation of the M phase until the genome has been replicated completely and any damage repaired. So in the G2M phase, what is checked? It is checked whether all the DNA has been replicated, whether there is any damage, whether if there are any DNA damage, whether that has been repaired or not. Okay. Then in the spindle assembly checkpoint, what is checked? Whether the chromosomes have been aligned, and they are associated with the mitotic spindle, and this will ensure that the complete set of chromosome is distributed accurately to the daughter cell. So each checkpoint, so this is an important point, so each checkpoint is controlled by mutual interaction between cyclin and cyclin dependent protein kinase. So there are different CDK and cyclin. So you do not need not remember the name of this CDK proteins and cyclin for, for this course, but yeah, um, for your interest, you can refer to the books and study about them. So there is cyclin and CDK protein. So the cyclin levels are regulated in different stages of the cell cycle, while the CDK protein, uh, the levels of CDK protein remains constant, and depending on which cyclin associates with the CDK protein, it controls which step will be blocked and which step will be proceeded. Okay. So now answer this question. Non-proliferative multicellular eukaryotic cell enter the dash phase. So the cells which do not divide further, they move into which step? Into which phase? No, G naught. Okay. So the G naught phase is also known as the quiescent phase. So any cell which is not dividing, it moves into the G naught phase. Okay. Now to answer this question, cell cycles at different steps are tightly controlled by cell cycle checkpoint. Each checkpoint is controlled by the mutual interaction between cyclin and cyclin dependent protein kinase. Okay, so there are different CDK4, CDK6, all these there, CDK8. Okay, now this is the last section that I will discuss today, apoptosis. So apoptosis, you all know about apoptosis. It is programmed cell death, right? Programmed cell death. So so when will this take place, and why is it required? So apoptosis will be required, for example, in our body. If we in our body there are millions of cells which are forming, and then they are again getting they are dying. Okay, so the natural process. So the natural process of a cell. So a cell is generated then after a period of time. So if you look into ribosome, it has a lifespan of 120 days. Okay, so after 120 days, it is broken down, and it is degraded. RBC, it is degraded. So all these, for these different processes, apoptosis is required, right? Then there can be any cell. For example, there is any virus infected cell. Okay, So if there is any virus infected cell, if that cell has to be, if you want that cell to die, then what process will take place? Apoptosis will take place. Then there can be a tumor cell which has been generated because of DNA damage. Okay, so there are several types of cancer cells which are getting generated in our body daily, and we want that such cells should not stay in our body and they should be removed from our body. So all these different so all these things, all these different processes will require apoptosis to take place. Okay, so what happens in apoptosis? So in apoptosis, if you look at the process of apoptosis, so in the first step, what happens? The chromatin starts condensing. So you see here, the chromatin will start condensing, and the nucleus. What happens? The nuclear lamina. I hope you remember the function of nuclear lamina. The nuclear lamina is present in the inner to the inner nuclear membrane, and so this nuclear lamina actually gets degraded, and once this nuclear lamina is degraded, the nucleus gets fragmented, and this fragmentation of the nucleus leads to fragmentation of the cell, and fragmentation of the cell will result in very small vesicles that are produced. These vesicles are called the apoptotic bodies. Okay, so apoptotic bodies are produced, and then these apoptotic bodies gets digested by the phagocytic cells in our bodies, like macrophages and other phagocytic cells which are present. So these cells will digest these apoptotic bodies and will it will lead to the death of the cell. Okay, so it is also called as, it is, why is it called as programmed cell death? Because a sequence of events take place which takes place in a very programmed manner. So first the chromatin gets decondensed, then there will be fragmentation of nucleus, then apoptotic body. So it is taking place in a very programmed manner. So it is called programmed cell death. In this, what happens? Chromosomal DNA gets fragmented. The chromosomes condense. The condensation of chromosome is known by a process called pycnosis. Okay, then what happens? The cells shrink. The You see here, the cell has started to shrink, or its size has started to decrease. Then but in so there are two processes, apoptosis and necrosis, and the difference between necrosis and apoptosis was discussed in the lecture, so you need to understand the difference between the two. So in necrosis, what happens? The cells swell and lyse. So one important difference between apoptosis and necrosis is that in apoptosis there is no fragmentation of the plasma membrane. The plasma membrane does not get damaged. Only this cell starts to shrink in size. But in necrosis, what happens? This plasma membrane gets fragmented, and once this plasma membrane is broken, the contents which are present outside the cell, they move in, and as a result, the cells swell, and once the cell will swell, it will lead to the lysis of the cell. Okay, so that causes the release of the content into the extracellular space. And one of the important proteins which are involved in this apoptosis process is the caspases. So caspases are the executioners of programmed cell death. So in caspases, caspases are a type of protein. Why are they called caspases? Because they have cysteine residues at their active site, and they cleave aspartic residues in the substrate protein. So if there is a substrate protein that has to be degraded, so this substrate protein will have an aspartic residue, and this caspases have cysteine residue at their active site. Uh, so uh, this when this caspases will act on this protein, uh, it will activate that protein to cause subsequent or activate further cascade of reactions. Okay, so these caspases are synthesized as procaspases, and it is converted to active form by other caspases by proteolytic cleavage. So procaspases are converted into caspases by other caspases. So there are caspase 3, then procaspase 3 will be converted into caspase 3, then caspase 3 will act on procaspase 8, it will convert it into caspase 8. So in this way, a cascade of reaction takes place. Okay, so there are initiator caspases and executioner caspases. So initiator caspases will begin the apoptotic process. It remains inactive, and what it does, initiator caspases will activate the executioner caspase. Initiator caspase, what will it do? It will activate the executioner caspase, and this executioner caspase will then further act downstream to activate the target protein molecules. Okay, so one method of apoptosis is the intrinsic or mitochondrial pathway of apoptosis. So this is intrinsic. What do you understand by intrinsic pathway of apoptosis? It means that any signal which is generated from inside the cell. So for example, there is DNA damage that can act as a signal for apoptosis of the cell, or there is a virus infection in the cell, then that will signal the cell to die. So all these this is called the intrinsic pathway of apoptosis, and it is also it is called the mitochondrial pathway because this pathway, it starts in the mitochondria. So what happens? So any there can be any signal for cell death, and this the cell death signal, it will activate pro-apoptotic proteins like BAX and BAC. Okay, so these backs and back, what do they do? Once these proteins get activated, these proteins form pores in the outer membrane of mitochondria. So you see these yellow colored proteins. So these are the backs and back complex. These form pores in the outer membrane of mitochondria. And once these forms pores in the outer membrane of mitochondria, the cytochrome C, these blue colored molecules you see, these are the cytochrome C molecules. These cytochrome C molecules get released from the intermembrane space to the cell cytosol. Now, once this cytochrome C is released into the cytosol, this cytochrome C forms a complex. It forms a complex with APAF1. This is another protein, and together this cytochrome C, APAF1, and caspase 9, it forms a complex called the apoptosome. Now, this apoptosome, what will it do? It will activate the procaspase 8 and cause the this procaspase 3 will be converted into caspase 3. Now this caspase 3 is active, and this caspase 3 will ultimately cause apoptosis of the or cell death. Okay. Once this caspase 3 is activated, then subsequently the nuclear lamina will get degraded. The chromatin, the different proteins which are involved in chromatin condensation, and those they will get activated. So the chromatin will start condensing, and ultimately the it will lead to the death of the cell. Okay. So yeah, so the cell death signals induces apoptosis. So remember these proteins, BAX and BAC, and another protein you remember is this is a molecule cytochrome C. Cytochrome C is an important molecule which when released from the mitochondria activates the intrinsic pathway of apoptosis. Okay, you tell me where it is found. We discussed in last class. Cytochrome C. You remember the electron transport chain? In the electron transport chain, the cytochrome C was involved in transferring the electrons from complex 1 to complex 3. So it is actually present in the inner membrane. It is actually present on the inner membrane of the mitochondria. Mhm. Hmm. Mitochondria. So that gets released from the intermembrane space to the cytosol. Okay. So if you go through the slides, then yeah, you can see in the ETS the electron transport chain, you will see the cytochrome C molecule. Okay, so the cell death signal induces apoptosis as a result of damage to mitochondria. Okay, so when active, what will happen? The pro-apoptotic effector proteins BAX and BAC, they form oligomers in the outer membrane of mitochondria. So once oligomers are formed in the outer membrane of mitochondria, it will result in pores. It will cause pores in the outer mitochondrial membrane, resulting in the release of cytochrome C from the intermembrane space. Okay, in the side from the intermembrane space, and now the release of cytochrome C results in the formation of apoptosome. Apoptosome is a complex which is formed by APAF1, caspase 9, and cytochrome C. Okay. Now, once this caspase 9 is activated, it will activate procaspase. It will convert procaspase 3 to caspase 3. So this is the effector molecule, and this caspase 3, once it gets activated, it will result in cell death. Okay, so this is the mitochondrial pathway of apoptosis. Now there is this extrinsic pathway of apoptosis. So this was discussed in very detail. So just I will cover the important points. So there is this TNF alpha pathway and this fast ligand. Okay, so what do you understand by extrinsic pathway of apoptosis? So if there is any signal from outside, then it is called the extrinsic pathway of apoptosis. Okay. So it is initiated by extracellular signal proteins. Extracellular signal proteins, like in this case, like in cytotoxic T cell. In cytotoxic T cells have this fast ligand. So this fast ligand, this is an extracellular signal protein, and on the target cell, there is a fast receptor or the fast death receptor. So this cytotoxic T cell having this fast ligand will bind to the fast receptor which is present on the target cell, and a cascade of reactions will take place one by one. So, initiated by extracellular signal protein, it binds to the cell surface death receptor. So, the target cell will have the fast death receptor, and once the fast ligand, which is present on the cytotoxic T cell, binds to the fast death receptor death receptor, then what will happen? Ultimately, what is happening is this caspase 8. So in that case, caspase 9 was getting activated. In this case, caspase 8 is activated, and this caspase 8 ultimately converts this procaspase 3 into caspase 3. So another similarity between the intrinsic and extrinsic pathway is that in both the cases caspase 3 is produced. Okay, in both the cases caspase 3 is activated. So but prior to that, in extrinsic pathway caspase 8 was getting activated. In the intrinsic pathway caspase 9 was getting activated. Okay, so once this caspase 3 is activated, the apoptotic substrate, they get activated, and ultimately it will result in cell death. So different substrates which are inactive, all these will get activated ultimately. So now answer these questions. So the role of pro-apoptotic proteins like BAD and WIM is inhibit caspase activity, promote mitochondrial permeabilization, activate DNA repair enzymes, and prevent the formation of apoptosome. So pro-apoptotic proteins were like this back and back. So yeah, similar to back and backs, these are also the another type of pro-apoptotic proteins, BAD and beam. So what it ultimately does, it will cause mitochondrial permeabilization so that the cytochrome C gets released into the cytosol, and subsequent steps can take place. Okay, so answer is option C, correct. Option B, yeah, correct. Now answer this question. The role of cytochrome C release from mitochondria in the intrinsic pathway is activates cas. Okay, option C. Yeah. So activates caspase 8. This is wrong because caspase 8 is gets activated in the extrinsic pathway. Okay. Inhibits caspase activity. This is wrong. It activates the caspase activity. It was activating the caspase 9. And third option is correct. Forms the apoptosome and activates caspase 9 and stimulates DNA repair enzyme. So this is also incorrect. So the cell will die. Why will it try to repair the DNA? Okay, cell will die. Answer is option C. The role of death repair receptors in the extrinsic pathway of apoptosis is inhibit apoptosis. This is wrong, right? So tell me the answer. What are the options? Inhibit apoptosis, activate caspase 3 directly, induce mitochondrial permeabilization, initiate signaling cascades leading to apoptosis. Yeah, option D. Option D is correct. Option D is correct. Why? Mm-hmm. Yes, yes. Yes, absolutely. So mitochondrial permeabilization is induced by this Bax and back protein. Okay, Bax and back protein. So this death receptor has no role in that. Death receptor will be activated by this fast ligand or the TNF alpha protein which are present. Alpha receptor, those. Okay. So yeah, option D. Now Bax by BCL2 ratio in apoptosis. So this is one thing I didn't discuss. Actually, BCL2, BCL2 backs. So there are, if you have gone through the lectures, you must have have learnt about this pro-apoptotic and anti-apoptotic. What are the pro-apoptotic protein molecules and what are the anti-apoptotic protein molecules? So BAX is a pro-apoptotic molecule, while BCL2, BCL2, BCLXL, BCLXL, these are anti-apoptotic. These are anti-apoptotic molecules. So and these BAX, BAC, BID, BIM, all these are pro-apoptotic molecules. So the relative amount of the pro-apoptotic and anti-apoptotic or the signaling molecules will decide whether the cell will go for apoptosis or whether apoptosis will be inhibited. Okay, so that is why BAX is actually, just now we learned about BAX. BAX is pro-apoptotic. Okay, but remember that BCL2 is anti-apoptotic. It prevents apoptosis. This is anti-apoptotic. This is pro-apoptotic. So a ratio between this pro-apoptotic and anti-apoptotic, what will it do? It reflects the balance between pro-apoptotic and anti-apoptotic signals. This is correct. It indicates the rate of DNA replication. No, this has nothing to do with DNA replication. It measures the caspase activity. This is also incorrect. And it quantifies mitochondrial permeability. That is also incorrect. So basically, this regulates the balance between pro-apoptotic and anti-apoptotic signals. Okay, so answer is option A. Okay, so that's all. Okay, so now do you have any doubts? So this is one of the slides. Yeah. Mhm. Yeah. Mhm. Uh-huh. Uh, yeah. So caspase is, so what happens? So the main players of apoptosis are the caspase enzyme. ठीक है? ये एक तरीके का एंजाइम है. So दिस एंजाइम, इस एंजाइम में एक एक्टिव साइट रहता है. तो जैसे नाम है ना इसका कैस्पेज, तो ये सी आता है कि सिस्टीन, इसके एक्टिव साइट में सिस्टीन रेसिड्यू रहता है. ठीक है, सिस्टीन रेसिड्यू रहता है और ये जिस प्रोटीन पे बाइंड करता है ना, या जो भी मॉलिक्यूल को, जैसे न्यूक्लियर लैमिना के कोई प्रोटीन को डिग्रेड करना हुआ, ये क्रोमैटिन कंडेंसेशन में कोई प्रोटीन इन्वॉल्व है उसको डिग्रेड करना हुआ, तो वो स्पेसिफिकली क्या करते हैं, उस प्रोटीन के जो एस्पार्टिक एएसपी जो है ना, कैस्पेजेस का नाम इसी से आता है, सी फॉर सिस्टीन एंड एएसपी फॉर aspartate aspartate residue. The aspartate residue is present on the target protein molecule. ठीक है, जो टारगेट प्रोटीन मॉलिक्यूल है उस पे एस्पार्टिक रेसिड्यू के साथ, जो ये एस्पार्टिक रेसिड्यू है उसके साथ बाइंडिंग करके फिर इस बाइंडिंग से वो उसको डिग्रेड करता है. ठीक है, या एक्टिवेट करता है. या फिर जो ये है ना, जैसे ये सिस्टीन रेसिड्यूज आर प्रेजेंट इन देयर एक्टिव साइट्स एंड क्लीव एस्पार्टिक रेसिड्यूज इन द सबस्ट्रेट प्रोटीन. सो एक कैस्पेस, वन कैस्पेस इज एक्टिवेटिंग अनदर कैस्पेस. जैसे हमने अभी देखा ना कि कैस्पेस 3 जो था, वो कैस्पेस 8 जो था वो कैस्पेस 3 को एक्टिवेट कर रहा है. तो वो कैसे करता है? सो द कैस्पेस 8 एक्ट्स ऑन प्रोकैस्पेस 3. जो प्रोकैस्पेस 3 है, प्रोकैस्पेस 3 गेट्स कन्वर्टेड इनटू कैस्पेस 3. ठीक है, प्रोकैस्पेस 3 गेट्स कन्वर्टेड इनटू कैस्पेस 3. हाउ? So this gets converted into caspase 3 by तो इसका कुछ पर्टिकुलर अमीनो एसिड स्ट्रेच है जिसमें एस्पार्टेट रेसिड्यू है. वो पर्टिकुलर रीजन क्या होता है, वो चॉप कर देते हैं, उसको कट कर देते हैं. तो जब ये कट हो जाता है तो ये एक्टिव हो जाता है. ऐसे होता है. ये स्पेसिफिकली एस्पार्टेट रेसिड्यूज जो है ना सबस्ट्रेट प्रोटीन, मतलब मान लो कि कोई प्रोटीन है एस्पार्टेट, फिर इसके बाद मान लो लाइसिन, फिर ग्लूटामेट, फिर एलानिन, ऐसे है. तो यहाँ पे कुछ ऐसे स्ट्रेच है, तो ये एस्पार्टिक रेसिड्यू पे बाइंड करके इस पार्ट को क्लीव कर देगा और Now, because this part gets cleaved, now this protein will get activated. ठीक है, जैसे और भी पढ़े होंगे इसमें, लाइसोजाइम में, फिर एंजाइम कैटालिसिस में पढ़ोगे. We will also learn about those how these different sites, this this cysteine protease, serine protease. There are different types of proteases are in the cell, right? Serine, cysteine. So this is a cysteine protease actually. ठीक है. हाँ, क्लीव कर देगा. हाँ, और क्लीव होने के कारण से वो एक्टिवेट हो जाएगा, फिर वो दूसरे को एक्टिवेट करेगा, ऐसे करके कैस्केड. हाँ, अलग अलग मतलब ऐसा नहीं कि आ, कि वो पूरा ऐसे डिसोसिएट कर देगा, उसका पूरा तोड़ देगा, ऐसे नहीं. वो एक पर्टिकुलर रीजन पे ही बाइंड करेगा और उस रीजन को रिमूव कर देगा, जिसके कारण से वो एक्टिव फॉर्म में आ जाएगा. जो इनएक्टिव फॉर्म था ना, प्रोकैस्पेस जैसे हो गया, वो इनएक्टिव था. देख, ये जैसे ही क्लीव हो जाता है ये एक्टिव हो जाएगा. ऐसे कुछ कुछ एक डोमेन. हाँ, चेंजेस होगा. तो ये एक तरीका होता है कुछ भी एक्टिवेशन करने का, ये भी. ठीक है. Okay, so we will take up more doubts in the next class. So I hope सब समझ आया है ना. Okay. 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 Okay, thank you.